Hey everybody, so as I mentioned, I'm a postdoc right now at the Wild Salmon Center, and I'm part of a really big project team. Working on this kind of really zoomed out 10,000 view study, looking at Chinook salmon throughout all the different habitats that they use, from the freshwater side to the ocean, and then also looking at how they are doing in all these different tributaries. This is a project that's a NIFWIF funded project that was identified as a research need by the U.S. Fish and Wildlife Service. And so we've been having conversations with folks at the Yreka Fish and Wildlife Office to kind of understand the research needs and research goals for this project. So for this project, we didn't collect any additional data. This was purely a synthesis project. So a project like this really wouldn't be possible without all these different entities and people working at all these different groups collecting this data. And I know a lot of folks are in this room or also online. So just thank you for all the contributions to this project. So this project really came about because Chinook salmon in the basin have really had a prolonged period of extreme low abundances that started primarily around the mega drought period that happened a decade ago, and then also conditions in the ocean around that time were also really poor. And so since then, the population really hasn't recovered since then, and it's been listed as overfished since 2018, which means that the population average escapement has been below this minimum stock size threshold. And actually, in the last 10 years, the escapement has fallen below this minimum stock size threshold 8 out of the last 10 years. And in the past, historically, we've had periods of time in which the escapement was also pretty low and below this threshold. But I think what's really unprecedented about this time is the lack of recovery, and this prolonged period of low abundance is really something that we haven't seen before. So that really drives the impetus for this study, which is looking at factors that have been suppressing the recovery of Chinook salmon in this basin, and looking at environmental factors both on the freshwater side and also in the ocean that have been preventing the recovery of Chinook salmon. Since Chinook salmon are not just found in the main stem or in one river, they're found in there's multiple different populations that are part of this complex. We were also interested in comparing the productivity and survival and dynamics between all these different tributaries to look at some key recovery focuses going forward. So this analysis that I've been working on in the last year is really broken down into 3 components that are tied together. So first, it's looking at the adults coming back and their red survival and fry survival until the juveniles begin out migrating from their respective tributaries. The second component is looking at, once they move from the tributaries into the main stem, what is their survival like as they're moving downstream into the ocean. And then the third is once they're in the ocean, what is their survival like as smolts when they first hit the ocean, and then once they become a larger size, how much are they impacted by fisheries, both in the ocean and then also in river when they return to spawn. So just going to jump right into the first component, which is looking at the tributaries or freshwater productivity. And so for this component of the analysis, we were looking at spawner escapement abundances from all the different monitoring programs throughout the basin and different methods that are used, and the respective juvenile migrant abundances from all the different monitoring programs and trapping sites. And in terms of freshwater productivity, the way that we were looking at this is total juvenile biomass per spawner upstream of each trap. So not just looking at the number of fish at each site, but also the size and the weight of these fish as they're passing through. And we were able to get data from folks from all across the different sites in the main stem, in the Trinity, and then also in a lot of different tributaries to look at freshwater productivity. So some of the factors that we were interested in looking at was spawner abundances, whether that affected the productivity rate. So this is looking at density dependence and whether there's lower productivity at high abundances. Looking at, I think this is a little cut off. Looking at spawning and incubating temperatures and flows. So we looked at spawning flows during the time in which adults are coming back and spawning, and then temperature and flows during the spawning and incubation period. So looking at maximum flows during this time period, but then also average flows during this time. And then once the fish begin emerging and moving downstream, we also looked and see at whether there were effects of outmigration temperatures and flow on productivity. So this is kind of just an initial results from looking at productivity. It's a little cut off at the bottom, but I think the last one is just outmigration temperature. And so I'll just point out kind of some of the key takeaways from this chart here. So for the spawners abundances and almost all of the different screw traps, we see a negative effect of spawner abundance on the productivity rates. So this is evidence of density dependence at almost all the different screw traps. We don't see an effect of density dependence at Bogus Creek, which was kind of surprising for me, and that's still something that I'm looking into. And then I'll just point to here that this is showing spawning and incubation average flows and seeing a positive effect of flows. And then for temperature, the mean temperature, depending on the average temperatures in each of the tributaries. For some of the tributaries, such as the Scott River and the main stem, warmer temperatures actually resulted in higher productivity, partially because these rivers are pretty cold during the winter time, so more warm temperatures increase productivity, but we saw negative effects of temperature in other sites too, and these are places that we think that are more bumping up closer to the optimum temperature, so warmer temperatures are going to result in lower productivity. For spawning and incubation flows at maximum flows, we also saw a negative effect of maximum flows in Shasta, Bogus, and at the Willow Creek trap, which suggests that there's high flow events that could be affecting the red and juvenile survival. This is basically showing the size of fish as they've been leaving the different tributaries in all the different years. And I'll just point out that for the most part, it seems like for all the different tributaries, the size of fish leaving overall have remained pretty similar in the most recent years compared to the earlier years of our study. But for the Iron Gate releases here, so the top here is the fingerling releases during the springtime and then the yearling releases in the fall. There's been fish have been released at a smaller size in the more recent years than in the past. This is in large part because they're released earlier, but even accounting for size and age, they've been smaller in more recent years. Once the fish begin moving from their respective tributaries into the main stem, for this component, we were playing around with the S3 model or the stream cell modded simulator. And this basically simulates the movement of fish downstream. So you can enter how many fish are starting out at the top of the river and simulate their mortality as they're moving down. This model also has a growth component, a bioenergetic model, so that you can also track what the expected size of fish to be once they reach the mouth. And what's really useful about this tool is that there's also a disease subcomponent, so we can also include data on spore concentrations in the river and temperature-dependent mortality from sea Shasta to model how much mortality might be happening because of sea Shasta. So this is one of the outputs from the S3 model, basically showing what we expect with sea Shasta mortality to have been since we have had this spore concentration data. And you can see that for the most part, the upriver populations like this year's Bogus Creek and the Iron Gate releases during the springtime, Scott River and the Klamath River main stem are all significantly affected by sea Shasta when they happen. Interestingly enough, on the bottom here, this light blue here is Shasta River fish, which are also an upriver population that swim through this area in which we know sea Shasta exists. But because they're heading out at an earlier period of time when the water typically is more around 15 or 16 degrees, the model estimates their mortality to be a lot lower. And this is especially interesting compared to the Scott River, which is in the model further downstream and actually not swimming through a large portion of the area of infection. But because they're heading out a lot later and a smaller size, their mortality rate is actually estimated to be greater than Shasta River fish. So from this S3 model, we're also able to estimate the size of fish from all these different tributaries in which they reach the mouth. And overall, across all these different populations for both the natural origin tributaries and also for hatchery fish, we see and we expect there's been a decline in the size of fish in which they're reaching the mouth. And this is partially because the conditions in the river have poor conditions for growth in more recent years. But a large part of the reason why the fish on average are been smaller is because they've been reaching the mouth at an earlier date. And so so this is showing the date of arrival. So this kind of upward trend that we see in a lot of different sites is basically showing that the average time in which fish have been entering the ocean is expected to be earlier in the more recent years. And this isn't so much, I don't think this is so much that fish are moving more quickly because of warmer conditions, although I think that is happening. I think a large part is that the later emigrating fish are not surviving. So in years in which we have poor summer conditions, we have basically a wall or this filter that fish that are migrating down are hitting, and the later migrants are not surviving. So in the past, we might have had this pretty large range of fish migrating from the early spring to the summer, but in more recent years, it's basically has been a wall that fish after a certain period of time in June and July are not surviving, and this is going to lead to an average earlier shift in terms of the average fish that do survive and arrive to the mouth. And this has major implications in terms of what we think their early ocean survival might be when they first enter the ocean, because we know size and timing are really important for determining the early ocean survival. And so what does it mean if in more recent years it's those early migrants that are the ones that are surviving? So we were able to look at this and look at what the early ocean survival has been for fish in their first year. And so this is the survival of the smolts within their first year of life in the ocean, and it's generally somewhere between 2 to 15% in the first year. And here I also have we also have estimates of early ocean survival for Redwood Creek, which is the adjacent creek south of the Klamath River. And what's really cool is we see pretty strong correlation both in terms of the year-to-year variation, but also the absolute estimates of early survival rates. So that for me was just a good gut check on the accuracy of these early survival estimates. So now we're kind of looking at what are the different predictors or what environmental factors in the ocean that we see that are correlating strongly with salmon survival in the ocean. And this is something that we're kind of actively working on now and testing different predictors And so some of the different predictors we're looking at is the size of fish and the timing in which we estimate that they're reaching the mouth, whether that has an impact on their survival rates. We're also looking at regional indices such as PDO and MPGO, and this Northern California temperature and upwelling index here. And then not just looking at kind of regional processes, but also just local temperatures and upwelling right near the mouth. And because we think that these fish are hanging out near the mouth once they enter the ocean. And so what's the habitat like in the more recent years for these fish locally? And then what's really awesome about studying Chinook salmon from the Klamath in the ocean is that we have a really unique data set right nearby us off of Trinidad, collected by Cal Poly Humboldt and NOAA on various oceanographic conditions, including krill availability off in the ocean. So we're able to directly assess whether food availability has had an impact on salmon survival. And so this is something that we're kind of actively working on now. So I don't have any figures on this, but I'll just point out that some of the things that are coming through right now is it actually seems like the this MOSI index here, which is an index of productivity and upwelling conditions in Northern California, has been a strong predictor for salmon survival. And then the other thing that we're finding as a strong predictor is juvenile krill densities during the winter time. And so when salmon first enter the ocean during the summertime, this is a period when they're feeding and growing a lot, but in the wintertime, they're really limited on food and might starve. And so we're finding that food during the wintertime has been a really important piece of salmon survival in the ocean. So that's kind of just a quick summary of kind of some of the key findings that we're finding in this study. And I'll just summarize that really we're seeing a huge kind of trickle down effect of these poor summer main stem conditions on survival. And it's really impacting kind of the peak and the latter half of the emigration that's happening during the summer. We're seeing that it's the main stem effect is the main stem conditions are really affecting nearly all the different populations, but a really unique exception is fish from the Shasta River, partially because they're heading out earlier while the river is still pretty cool. And then this poor summer conditions are almost acting as a filter on the population, and this is likely going to result in a shift in terms of the timing in which fish are reaching the ocean. And so fish that are the ones that are moving earlier and also smaller are the ones that are more likely to survive during poor summer conditions compared to the peak migration period. And historically, we've thought that these early and smaller individuals are thought to have low survival and not contribute a large piece to the returning population. And so now, if potentially these are the fish that are the ones that survive, it's really important to support these early migrating fish. So that's kind of leading into some of the conclusions that we have, that these early migrating fish might be the fish of the future, or the fish that do survive to the mouth when the river conditions are poor. So it's really important to improve the downstream habitats for these early migrating fish. And then for the fish that are migrating during the peak season, during the summertime when it's warm, refugia is going to be really important as these fish are moving down. And I didn't really get a chance to talk about the fishery side of things, but we have a lot of interesting results on the fishery side. Folks want to talk to me about that afterwards, but I'll just share some of the conclusions from that piece of the analysis. And we're seeing changes in the population structure and how fishing effort is translating into fishing impact. And so for the ocean fishery side, having this conclusion that it's important to respond to the changes that we're seeing in the population structure in the ocean. So I'll just end there by again thanking everybody that has been a part of the data collection process for the synthesis project. And especially for these individuals for being point of contacts and for people to talk to about this project.
I was just wondering where your ocean survival was coming from, as what that number going into the model came from. Like an estimate based on the total number out of juveniles, and then what's coming back and trying to figure out the composition. I'm just wondering where you're getting ocean survival.
Yeah, I had an awesome figure diagram for that I had to cut out, but yeah, basically it's we were able to take all the different ages that age composition and like convert that back to the cohort in which they were born in. And then we were also able to subtract out fishing mortality and natural mortality that happens to these older individuals, and that's how we were able to narrow it down to survival during that first window of time. Yeah.
Could you tell if I think you said the late out-migrating fish are not surviving as well? Can you tell if that is a sort of a climate change driven thing that's likely to get worse, or a fish disease, sea shasta thing that's likely to get better?
I think it's both. So in the S3 model, there's the disease component and there's also temperature dependent mortality. And so if in the model, if temperatures exceed 20 degrees, there's a additional mortality rate that's applied. So I think in more recent years, it was both that was happening. And also in terms of sea shasta mortality, that's also tied to temperature. And so I think temperature kind of has a double effect, like one effect just on warm temperatures, and then one effect on increasing the mortality rate from sea shasta. Yeah,
I just have one comment. So on the Scott and Shasta traps, we have like a 20-year data set for out-migrants there. And I've always noticed this small bump in a one flush Chinook life history that leaves the system very early. So it's staying through the summer and then leaving, sometime in January. And we're trying to expand our monitoring to capture more of that, but it was just interesting to see that could play into all of this, even though it's not a large piece of the population. They are leaving at a time when water quality conditions are better in the river.
Mm-hmm. Yeah, totally. And yeah, it's again, that life history is super difficult to keep track of, and but it would be yeah, interesting to think about how with climate change and changes in the environment, how these different life histories are going to be in the future. Oh,
We have time.
Mike. Go ahead, Mike.
This is Mike Orchid, the Hoopa Valley tribe. It's a small matter here, but right on the cover here, you got our name on there, and I would note that I believe I heard about a year ago about this whole study from Michael O'Farrell, who's with NOAA Fisheries, and I might have also talked to Bill Pinnix about it, but we haven't actually been actively involved in it. I mean, the data and information, and certainly the Trinity River Restoration Program is a major focus of the Hoopa tribe, and so that's one thing. I can get over that, but the other is I think we really need to we get in this compartmentalized world here, and I think there's so many overlaps here. I can tell you a couple of them, and it's and maybe talk offline with you if you're interested. The Pacific Council is undergoing a process for a management process, which data and information potentially from your study could be very valuable in terms of productivity. And then narrowing down to the Trinity, at times the Trinity produces upwards of 50% of the fall Chinook production presently in certain years. And so the restoration program there is working on a limiting factors analysis And there's a lot of overlap with the people there, and you could say, oh, that's supposed to occur, but I'm from Missouri there. I don't necessarily fully agree with that. And so I just think there's some real opportunity there to cross lines there. One of them is a direct management output that the Pacific Council could do. The limiting factors analysis for the Trinity Restoration Program is long overdue. Not long overdue, but it's something that's been worked upon, and the tools and everything have taken a long time to develop. And so I just think it would be a really, really strong addition to your work there. And so I'll end there, and thank you for your presentation. Otherwise,
Yeah, thanks, Mike. I have checked out the limiting factors analysis, but should definitely follow up afterwards.